Preparation of the extraction liquid. The extraction liquid is prepared in a beaker by mixing 20 ml of dishwashing liquid, 5 spatulas of salt, and 250 ml of distilled water. Then, we mix. Let's move to the strawberries. The strawberries are placed in a resealable bag. The extra air is pushed out, then the bag is sealed tightly. The strawberries are squeezed and smashed using fingers for 2 minutes. Next, 20 ml of the extraction liquid is poured into the resealable bag. The strawberry mixture is squeezed using fingers. The strawberry mixture is poured from the bag into the beaker through a strainer. It is allowed to drip into the beaker until very little liquid is left. Contents of the beaker are poured into the 50 ml tube. The tube is tilted and 15 ml of cold rubbing ethanol is very slowly poured down the side of the tube. The glass rod is dipped into the 50 ml tube at the point where the alcohol and the strawberry separates. The glass rod is pulled up and observed.
Let's make a cone.
The narrow sticks are cut into several shorter lengths and they are taped to one side of the regular sized cross stick. The teeth are ensured to be evenly spaced to hang down to the same level. Another regular sized cross stick is taped parallel to the first one so that the teeth are sandwiched between the two sticks.
The gel solution is made by adding 0.5 gram of agar-agar powder to 50 ml of the sodium bicarbonate buffer to dissolve the agar-agar powder. So the gel solution is heated in the microwave. So the solution is expected to become translucent and will solidify at room temperature. The solution should be washed carefully as it may boil over when hot.
Once the solution has cooled enough to be touched safely, it is poured into the box until the cone teeth are submerged by approximately 0.5 cm. Hooray! The gel has solidified! The bottom half of the gel and a thin strip from the top of the gel are cut away using a knife. At this point, the comb has been gently removed by being pulled straight upward without tearing the gel.
Each piece of stainless steel wire is length so that it fits inside the box along its width and hooks over the side. One electrode will be placed along the top end of the gel, other electrode will be placed at the bottom end. So a high voltage power supply is made like connecting 5 9V batteries together with the positive terminal of one inserted into the negative terminal of the next. Two batteries are clipped together using alligator tips.
The DNA sample are prepared by mixing the previously isolated DNA with 0.5 ml of glycerin in a small tube.
Once the gel and samples are ready, just enough sodium bicarbonate buffer is poured in to cover the solidified gel.
A pipette is used to transfer approximately 5 ml of each sample into an empty well.
After all samples have been loaded, the leads from the power supply are connected to the stainless steel electrodes attached to the box. The power is left connected for 15 to 20 minutes.
Power supply was off. A few drops of methylene blue solution are added onto the gel. The stainless steel wire is removed from the box. The box is gently agitated to allow even staining and the gel is left for 5 minutes.
From the image, the colored bands have all migrated downward, which means that the bottom of the gel was the positive electron. Most of the colored samples moved toward the positive end because they carry a negative charge. Therefore, they are attracted to the positive terminal when an electric current is applied. DNA has a sugar phosphate bond. Each phosphate group carries a negative charge. DNA molecules are strongly attracted to the positive electron. When current is applied, DNA will migrate from the wells, which is the negative end, toward the bottom of the gel, which is the positive end. Methylene blue moved in the opposite direction during electrophoresis. This is because methylene blue carries a positive charge, so it was attracted to the negative electron at the top of the gel.